I've been getting in trouble for disclosures lately, so I have to go through these. So I'm at Children's National. I direct the CINRG group, but it's elected. I'm chair-elected at TreatNMD, a great group through Europe. And then private, I'm co-founder and CEO of Reverogen Biopharma, which I'll talk about on Friday, and co-founder and VP of Agata Biosciences. And CINRG is spinning off a for-profit called TRINS, which will aid clinical trials and post-marketing in DMD. And that's coordinated with TreatNMD. Okay, so what I thought is, since we're in DC, we should bring up the roll call. That's our local sort of political newspaper here for Congress. And just a couple days ago, you're now a Beltway insider, if you haven't recognized that. You're in DC. So applauding the 21st Century Cures Initiative. And this is a commentary in this. And I just drug out a quote there, which I think is really nice. Sometimes you have to tip your hat for a job well done. That is the case with the 21st Century Cures Act. This is not something you hear about our government lately. The 21st Century Cures Initiative seeks to address a complex problem. Life-saving medicines, medical devices, and diagnostics take too long and cost too much to develop and bring to patients in need. So this is a really nice bill that was just unanimously passed by downtown, and there's really some good meat in that. And I'm going to use this as sort of an intro to the biomarkers part, and you'll see why in a second. So this is the 21st Century Cures Act, and it has 4 different sort of sections, so I'll just briefly go over those. So the titles, they're called. So Title 1 is Discovery, and it mostly deals with National Institutes of Health. But what you'll see is Duchenne is just written all through this language, which is really great, and I'm sure that Parent Project and many in the room had something to do with that. But promoting pediatric research through NIH, facilitating collaborative research, clinical trial data system, national neurological diseases surveillance system, that's us, data on natural history of diseases, that's us in here, patient-focused drug development, use of tools, FDA advancement of precision medicine, modern trial design, expediting patient access to all these new drugs, that's in there. Orphan drug product extensions, exclusivity, enabling FDA scientific engagement. You're seeing that. You have FDA talking at this meeting, which is terrific. And they're really trying hard to talk to everybody. And it's now legislated as well. And then the other 2 titles were delivery and Medicaid, Medicare, and other reforms. And I just talked to somebody just an hour ago who sort of seems to be involved in writing this. And while there's lots of debate about Medicare, et cetera, the rest of this stuff looks like it'll actually get passed, all the stuff we just talked about. So why do I bring this up? Well, if you look at the very top in Discovery, Title I Discovery, National Institutes of Health, they talk about innovation funds, strategic plan. The #1 topic mentioned in this bill is biomarkers. So that's what we're here to talk about this session, is biomarkers. They also talk about precision medicine, infectious disease, antibiotics, and other things we've heard about, but #1 is biomarkers. So drill down, what are biomarkers? Biomarkers is one of the most generic words invented in the English language. Technically, a plant growing, responding to light is a biomarker. Okay, so we're going to have to drill down and really define them. And I want to actually get very serious about this because defining biomarkers is critical. I mean, you really have to, because it's very disorienting, and I'm just going to step you through. There's like 4 flavors, okay, and you really have to keep them distinct because they have really different purposes. So that's what I'm going to do in the rest of this talk. So drill down, what are biomarkers? Well, the Act actually drills down for us, and they define these 4 classes. So the first one is responders versus non-responders, and it's written out here. The evidence needed to support the use of biomarkers that identify subsets of patients as likely responders to therapy in order to streamline the conduct of clinical trials. And I'll talk about examples specifically in Duchenne in each of these, but first I'll just go over the 4 categories. So the first one is responders and non-responders. The second one, which is a really important category, which we'll spend most of the presentation on, is predictor of drug response. So what they wrote, recommendations for the design of studies to demonstrate the validity of a biomarker as a predictor, and predictor is a really important word, and both John and Lee talked about it, of drug or biological product response. Lee just mentioned biomarkers as acute readout of drug response, something that you can detect, and it's objective because it's not subject to placebo effect. A biomarker, you can't hope to be better. So that's really important because I'll talk about that more later. Number 3, biomarker trials, the manner and extent to which a benefit-risk assessment may be affected when clinical trials are limited to patient population subsets that are identified using biomarkers. And then the fourth one, which is, they had two lumped together, I just called them companion diagnostics. And it's really using molecular diagnostics and different diagnostic tests to make sure you're treating the right patients. So now I'm going to drill down into these. These are the four categories: drug responders versus non-responders, predictor of drug response, and as I mentioned, that's a big category, many subcategories, but fortunately, we also just released last week, thanks to you guys, was the FDA guidance for Duchenne muscular dystrophy, and biomarkers is in that, and so we're going to draw from that language. Biomarker trials and companion diagnostics. So to get two of these quickly out of the way so I can focus on the meat of this presentation, let's just talk about biomarker trials and companion diagnostics, and that's all I'll talk about them. Biomarker trials, we at Children's Hospital just had a drug approved for urea cycle disorders. It was actually one of the new vouchers that was just sold for $250 million because of legislation. That was done on 2 patients, unblinded. You give the drug, you watch their blood change. That's all you did, and it was approved. Now, what were you watching? Well, urea cycle is a disorder where you can't metabolize a protein appropriately. So you may have, if you get too much protein in your body, you can get ammonia building up in your blood. It causes your brain to swell, and you can go into a coma, and you can die from an attack. Okay? Now, doing a trial on a bunch of patients to wait until their brain swells and they get an attack and a coma doesn't sound good. Right? Now, what else can we do instead? You can probably already guess it. Just measure ammonia in the blood. If the ammonia never goes up, and the drug keeps the ammonia really low, even when you're starting to eat protein, it should work. Right? You'll never get that ammonia causing. So that's what a biomarker sort of trial is. We really don't have that in Duchenne. It's maybe dystrophin in the future, but it's tough. So I don't think this is so relevant to DMD. Companion diagnostics, on the other hand, is really assumed in DMD. It's all over. I mean, you basically, to enter a clinical trial, you need a DMD gene mutation. That's companion diagnostics. Okay, so it's so obvious, I won't deal with that anymore either. It's really embedded in every trial that we're talking about. Okay, so now what I'm going to focus on is these two. A couple slides on responders versus non-responders, and then the rest of the presentation on predictor of drug response. So responders versus non-responders is most often in Duchenne is called genetic modifiers. Now let me go over what genetic modifiers, and this is work I'll be presenting from Luca Bello, who is a great neurologist who switches his time between Children's National here in Padua, Italy. The CINRG group, Craig McDonald, the natural history study, many of you perhaps participate in that. And we've also had a nice international collaboration with Kevin Flanagan, Weiss, Batali, and Pegararo, multinational, to do this sort of work. Okay, so whoops. We all know that about Duchenne. You've heard that in other things. All patients have the same biochemical defect. You're missing dystrophin, and generally have a similar clinical progression, or what's called a phenotype. But why isn't that working? I'm missing something there. But basically, it's quite variable. So what you see here, let me see if I can find it, is you see lots of variation with some mild patients and some severe patients. And this is, let me see. Oh, I'm missing a couple slides here. But oh, that was a slide in the other. Okay, so the pictures that are missing there is it's 2 6-year-olds. And you guys have seen this. I'm preaching the choir here. But you can take a 6-year-old with Duchenne, one who's very strong, and another 6-year-old with Duchenne who's very weak. They both have Duchenne. They both have the same mutation often, same missing dystrophin. So those are what we call genetic modifiers. They're differences between us and just anybody in the world, just polymorphisms that make us all individuals, that interact with the Duchenne muscular dystrophy to make that more severe or less severe. So there's been a lot of progress in that, and here they're just defined: polymorphisms unrelated to primary gene defect, modulate disease severity, onset, progression, response to therapies, identify important factors in disease pathophysiology. This is quite important because if we find a robust single-letter change out of 3 billion in the genome that dictates a mild versus severe Duchenne, well, a drug against that same protein should make mild versus severe Duchenne. We could make kids milder with a drug. So they're molecular targets for therapeutics. And then they're also important in clinical trials because one of the problems with seeing if a drug works is Duchenne can be variable in presentation and progression. Well, that makes a clinical trial and seeing drug effect more complicated. So if we can control for that and understand what's causing it. So right now there's 2 robust genetic modifiers, and then we collaborated a third one which isn't out yet. One is osteopontin, another one found by Kevin Flanagan and Elizabeth McNally, and is LTBP4. And that was a nice example of going from mouse to human. And I'll just show you some recent data that was just published. What do these genetic modifiers mean and what do they do? So in the natural history study that we've conducted, 340 DMD kids followed from 2 to 28, 7 years longitudinally, recruited at 20 centers worldwide, up to 7 years on disease milestones and functional outcomes. So what we did is use this sort of population to define what genetic modifiers do. And so here's what happens. So here's SPP1, osteopontin. And what you see is it's really a steroid responsiveness locus. Those patients with one genotype at this gene, so one version of this the SNP, it's called a SNP polymorphism, gives you a 2-year difference in response to glucocorticoids. So if you have one genotype, you'll do really well on glucocorticoids. If you have another genotype, you won't do so well on glucocorticoids. And it's 2 years of mobility difference. So 2 years later into losing ambulation. LTBP4 is not steroid responsive. It seems to be generally in the Duchenne population. Again, 2 years difference in loss of ambulation. But this one's a little rare. So only 10% of patients have that flavor of the polymorphism to become a milder patient. So these are the 2 genetic modifiers, and we can start thinking about those, stratifying clinical trials, and using those as drug targets. Now, just last 2 slides on genetic modifiers, and then I turn to the rest. One thing, most genetic modifiers have looked at the age of loss of ambulation. Well, that sort of limits the number of subjects you're looking at. What are all those kids who haven't lost ambulation? So what you can actually do is take things like strength, 10-meter walk, 6-minute walk, time to stand from the floor, and use computer programs to see how they predict later loss of ambulation. So we've done that, and it actually works quite well. So you can actually use these other measures in younger kids to develop a prediction of later loss of ambulation, and that's been done. The other thing that's really important in genetic modifier studies is what's called population stratification. This is the last complicated term. What that means is, a lot of us think we know our ethnicity, that we may be Caucasian or European or African or Latino. Well, it turns out, if you're, like, these are people who are kids, Duchenne kids, and their parents who thought they were Caucasians, and many of them are, but many of them aren't. So a lot of us don't quite have the ancestry we think we do. And it could be because you don't have the father you think you do, or many other reasons. Well, don't laugh. Do you know how many of you in this room don't have the father you think you do? Seriously. 1 out of 20. So look at your neighbor. We know that rate. Now, I always wanted to ask my dad that. So this is the collaboration we've set up between PADO, an Italian network with Elena Pecoraro, BioNMD with Spitali, and United Dystrophinopathy Project with Kevin Flanagan, where we all sort of validate each other's genetic modifiers. And then you can also look at heart involvement, cognitive involvement, and look for other SNPs that influence those. And this is just the people that worked on the genetic modifiers. So that's it for genetic modifiers. Now I'll turn into the last major one, which is predictor of drug response, big category, many subcategories. And let's look to the FDA DMD guidance, which drills down on this. So this is the guidance that was just released, I guess, a week or 2 ago. And Parent Project had a major role in that, FDA. So this is taking out quotes from that. So typically, drug development programs for dystrophinopathies should include early phase studies that are short in duration. We're running into a problem that, as you just heard, there's so many trials that do we even have enough kids to enter into these trials? And we also fully recognize how much of a burden it is on the patients and the families to do these trials. And if a trial's a year long with lots of visits, with lots of blood tests and biopsies, then it's really a pain. So one of the things that's so much hope for biomarkers is they'll help with that burden, that there'll be an objective acute readout, that you'll see a drug effect maybe after a month instead of after a year. And that signature in the blood will predict the later clinical benefit. Okay, and so that's what we're going to do in this last category. Such studies may evaluate effects of drugs on biomarkers to identify doses that are most likely to be effective. This is a key term, what's called dose ranging or dose finding. A lot of these drugs you're hearing about, most of us are guessing at which dose might be the best. What if we try a lot of doses in a small number of kids, just quickly, and look for that blood signature to show that we've found the right dose because that blood signature is telling us, that biomarker is telling us it's the right dose, and that are sufficiently tolerated to be studied in definitive trials of longer duration. So, and here is a key thing which I'm sure you'll hear throughout the meeting is accelerated approval. Basically, in dystrophinopathies, biomarkers that reliably reflect the health and amount of skeletal muscle at a biochemical, cellular, tissue level may be useful across the whole drug development process, and Lee even referred to them in mice. And there's 4 different subcategories of these markers: prognostic, predictive, pharmacodynamic, and surrogate endpoints. Now, surrogate endpoints is sort of the holy grail that Lee showed on his slide when he talked about biomarkers. And dystrophin's often cited as that surrogate endpoint, where if the therapy is meant to bring dystrophin back, and we know that dystrophin should improve later clinical outcomes, why not approve the drug, give accelerated approval, just on the dystrophin finding? Don't even wait for the clinical benefit. So that's sort of the holy grail, and it's not only for dystrophin, but for other biomarkers. So I'll talk about the last few slides, dystrophin as a biomarker, and then blood as a biomarker. So dystrophin as a biomarker is really out of just muscle tissue, and we're only talking about biopsies, and it's not many of the drug trials, just a couple of them like exon skipping, but there's also been a lot of discussion. FDA just ran a whole day-long workshop, many of you know, on dystrophin as a biomarker. I think your best view of this is what's called a pharmacodynamic biomarker. Pharmacodynamic means you're seeing a reflection of the drug effect in the muscle. So if you have an exon skipping drug, and it should be causing exon skipping, and that exon skipping should make dystrophin, if you see increased dystrophin in the muscle, the drug's working. Right? So that's a pharmacodynamic biomarker. It's evidence for mechanism of action, and you have examples of atalurin, exon skipping. Mechanism of action should be new production of dystrophin in patient muscle. You can use that for dose finding, dose ranging studies. Can you pick the dose that makes the most dystrophin? And then eventually, hopefully, this holy grail of surrogate biochemical outcome measure does increase dystrophin reasonably expected to predict later clinical benefit. Now, one of the key things, and again, this has been quite a bit of discussion on this, including the workshop that was just held. Before you even start, can you measure dystrophin accurately and reliably? If you can't, then it's hard to make a compelling case to the FDA that this is a good biomarker. And to date, no dystrophin replacement trial has met FDA criteria for considering dystrophin as a biomarker. FDA has rules. And when you do run a test, it should work, and you should be able to show that the test is reliable, sensitive, specific. And so far, none has quite met that bar. So the additional problem is you're introducing Becker dystrophin and not normal dystrophin, and that also complicates FDA's interpretation. But there's a new assay that Yetji Pathout and Christy Brown at Children's have developed, a mass spec assay. So here's the FDA guidance. You should be below these numbers for your accuracy of the test. And you see we can get below it there, right on the edge for low levels and high levels. And this is just a comparison to immunoblots that are much higher than that. So they're just not quite good enough. So now the last couple slides is biomarkers that are not based on dystrophin. So this is generally an accessible patient sample, so not muscle, it's not considered accessible, but instead blood, pee, or spit. You need natural history of biomarkers in DMD, so you need to know how each of these the signature of the blood changes as a function of age, disease severity. And you want to then take that sort of palette of biomarkers and graduate some, elevate them to a pharmacodynamic biomarker. That you're seeing it's reflecting something happening in the muscle and reflective of drug effect. So where do we stand? Just over the last year, there's great progress in this. There's now a natural history of 1,500 blood biomarkers defined in DMD, and many of these are cross-referenced, the mouse model. Yetji Pathout in the audience, and his team has done a lot of this work. Some of it was collaborative with PPMD and SomaScan. These are 2 key papers using 2 different methods. And let me just show you one or two examples of data from that. Now this is in the MDX mouse, but it's just, this is normal mice, and these are a whole series of blood biomarkers. And you see that when a mouse is first born, many of them look like normal in a mouse missing dystrophin. But you see quickly, you see all this showing muscle disease activity show up, but some of them disappeared even a couple weeks later and then don't show up again. Some of them stay high the whole disease. Some of them are actually higher in normal than they are in Duchenne mouse. Well, you see the same thing in Duchenne muscular dystrophy. This is the most recent collaborative SomaScan paper with SomaLogic and PPMD collaborative. And here is patient age, so going from 5 to 25 years, each of these panels. Here's the level of the biomarker. Here the blue is normal controls, so just kids brought in and tested. And these are all Duchenne kids at different ages. And this one you know very well, CK, serum creatine kinase. It's one of the first tests that was done. Well, you see at a young age, it's extremely high, which you know. But look how quickly it falls with age. And that's one of the reasons that you don't test CKs in carriers when they're older, because it falls in carriers as well. Well, what does that mean for testing a drug? Well, maybe testing a drug at this age, CK is a good biomarker, because you can see it go towards normal. Testing CK at this age is a pretty bad biomarker, because it's already close to normal. Now you see other biomarkers that are elevated in Duchenne through the whole disease, but they overlap with normal a bit. Others increase with age. This could be some of your fibrosis markers. Some are higher. Well, this was the opposite. This is low in Duchenne and high in normal. This is high in Duchenne and low in normal, all the way. No, it's the other way around. The red is Duchenne. I'm having slide panic. Need Yetrib up here. The red is Duchenne, so this is high in Duchenne, low in normal, and it's switched here. So it's actually low in Duchenne. Okay, so just in summary, next steps in biomarker, you need to make some of these pharmacodynamic biomarkers, and what's been defined is which ones respond to glucocorticoids, both for safety and efficacies, which ones respond to dystrophin replacement in both mouse, dog, and human. Once these pharmacodynamic biomarkers are defined, integrate them into drug approval pathways to build the compelling story. To say that, yes, we have a biomarker that is predictive, sensitive, reliable, and an acute objective measure that predicts later clinical benefit. You can then share that data between all the different drug programs. So the vision thing is that biomarkers become acute objective readouts of mechanism of action, dose selection, surrogate outcomes, leading to less patients in trials, shorter trials, de-risking of programs, as Lee mentioned, and more and better drugs quicker. So really, that's why it's number one in 21st century cures, to go back to that legislation downtown, is because to make drugs faster, quicker, cheaper, and better. So summary, just to quote a last slide from the FDA guidance, it is important that trials with clinically meaningful endpoints like strength and mobility include as many biomarkers as feasible to help establish the correlation between such biomarkers and clinical endpoints. Before studies are undertaken, sufficient analytical validity should be demonstrated, assessment of performance of the test, quality control measures, documentation of results, and you have to build this compelling story, which they call considered reasonably likely. Consider use of other biomarkers, such as those measured with MRI, and Lee just talked about that, very promising. And then just the acknowledgements, the biomarker work at our shop with Yetri's team was funded by NIH, U.S. Department of Defense, NIDR, which is National Institute of Disability and Rehabilitation Research, to the UC Davis shop with Craig McDonald, Foundation to Eradicate Duchenne, Clark Charitable Foundation, Parent Project, Action Duchenne, MDA, and the Duchenne Alliance, Michael's Cause, Pietro's Fight, Alex's Wish, Ryan's Quest, and Save Our Sons. Thank you.